Hello, and thank you for joining us. I'm Gwen Taylor, Senior Editor with Current Protocols at John Wiley & Sons, and I'm delighted to introduce today's webinar titled The Role of GCMS-MS in Metabolomics Experiments and the Exploration of Optimized Workflows. This webinar is being co-sponsored by Current Protocols and Thermo Fisher Scientific. Thermo Fisher Scientific is a world leader in serving science, providing analytical technologies, reagents, consumables, services, and software for cutting-edge scientific research to routine industrial applications. Current Protocols is the largest collection of peer-reviewed, authoritative, and regularly updated step-by-step research techniques and procedures available for life scientists worldwide. With 17 titles and over 16,000 protocols, Current Protocols is part of Wiley Publishers. During today's program, we encourage you to submit your questions throughout the event by clicking on the Q&A box at the bottom of your screen. Your question will not be seen by any of the other attendees, so please don't be shy about asking them. The webinar will be recorded and available for viewing in the next few days. We will send each of you an email with details of how to access the recorded webinar, along with a PDF of the slides and a customizable certificate of attendance. Now, it is my pleasure to introduce today's speakers. Dr. Vladimir Shuleev is a professor in the Department of Biological Sciences at the University of North Texas, and he directs the Shuleev Metabolomics Lab. Dr. Shuleev's research focuses on studying the biochemical, cellular, and molecular mechanisms of plant responses to biotic and abiotic stress. His research group is working to develop metabolomics technologies that apply high-throughput metabolite profiling and targeted metabolite analysis to study stress responses in microorganisms, plants, and animals. Dwayne Cardona is the Senior Application Scientist for the Thermo Fisher Scientific GC Triple Quadrupole Mass Spectrometer Applications Group. He is currently developing applications for metabolomics research in collaboration with the Shuleev Research Group. Dwayne has a diverse background in GCMS sample analysis and analytical chemistry. So now let's begin with a very warm welcome to you, Dr. Shuleev.
Thank you very much, and we'll start our presentation, which is entitled The Role of GCMS-MS in Metabolomics Experiments and the Exploration of Optimized Workflow, which is a collaboration between our lab and Thermo Fisher Scientific, try to develop an optimized workflow for the targeted MS-MS-based analysis for metabolomics. And if you really talk about metabolomics, it's a new area that's becoming more and more developed, and to complement other omics technologies along with genomics, transcriptomics, and proteomics, to be used in a framework of a systems biology to really understand the cellular behavior. And metabolomics is really as a technique which allows you simultaneous analysis of a range of metabolites or metabolome. And if we can define metabolome, which is done by Steve Oliver a while ago, used to work in University of Manchester, is a complete set of metabolites or low molecular weight intermediates, which are context-dependent, varying according to physiological, developmental, or pathological states of the cell, tissue, organ, or organism. And measuring all these compounds in the cells is quite a challenging task, because unlike proteomics and genomics, which really focus on several metabolites or types of metabolites, the targets for metabolomics is quite diverse, and we'll come back to that later, but it's really a large set of molecules which range from lipids, amino acids, fatty acids, sugars, and so on. And depending on really what question you want to answer, your metabolic targets can be different, and metabolome can be measured in different ways. You can be measured a set of metabolites, which is called targeted analysis. You really want to identify and quantify a set of predefined molecules that are important in your physiological process. Or if you really want to uncover novel ideas and build novel hypotheses, and if you don't have a good understanding really yet about your biological process, you can do metabolite profiling to try to measure as many or try to measure all metabolites in the cell through metabolite profiling. And this way allow you to screen for underlying mechanism of your biological process and identify molecular networks involved in these processes. You can also try to use metabolomics to define biomarkers or measuring, which is called process fingerprinting if you measure intercellular metabolites, or sometimes footprinting if you measure extracellular metabolites, to really define the patterns that correspond to your specific state, let's say for your disease states, stress states, that can be eventually used as your markers of the condition. And if you talk about really metabolomic experiments, there's several well-defined steps that you need to think about before planning and executing your experiment. When you design your metabolomic study, you really want to first put a question first. What question you want to answer? And determine what kind of technology, what kind of work you will pick up later on, and I will come back again later in a few slides ahead. But it's really the very important experimental design in the whole metabolomic study. And the next step is very important, and many people underestimate, is your sample preparation, because depending on your techniques to prepare samples, a choice of internal standards, your results may be very much different. So you really want to put a lot of attention in a sample preparation step to make sure you can get the best samples for specific technique you use. And then the next step is really data acquisition. It's really when you take a sample, run it through your instrument of choice, and it's multiple instruments that you can use later on, and then you acquire your raw data. And the next step is very important, again, in metabolomics, because it's determine, again, the quality of your data is data processing. And it's, in metabolomics, there's two steps of data processing that are important. One is really the raw data processing, when you take a data from instrument and try to extract as much information into that can be available later for multivariate statistical analysis. And the next step is really your process data can go through multivariate statistical analysis and data mining to really determine your biological output and formulate your hypothesis, answer specific questions you want to answer. And the data processing is an important step because it also allows you to associate your peaks that you see from your analytical instrument into your name of your compounds. And really, compound identification is critical for your further statistical analysis and data interpretation. And this is why metabolite annotation is a critical step for any data analysis. Again, if you go really to your experiments, so you can really define your experiment in three larger steps. The very first one is your experimental design and sample preparation, where you really, first of all, take your organism of choice, in our case, let's say Arabidopsis thaliana, which is a model for plant research, and you want to extract the set of metabolites you want to answer, and determine what group of metabolites you want to measure or determine if it's a global profiling, a targeted analysis, specific lipidomics experiments, or you want to look at amino acids. Your experimental design and sample preparation will be much different because you need to select the best appropriate technique for the metabolites of your choice. Is it a polar or non-polar, lipids, amino acids, and so on. And the next step, again, is a separation. And for your separation, there are multiple techniques you can use. And so you can generate your chromatograms, a set of chromatograms or raw data files that you'll then be processing. And again, the next step, which is the most important, is really to associate all these multiple peaks that you see in chromatograms with the particular compounds that you can name and quantitate. And after you name and quantitate compounds, you can generate your compound list with your raw intensities or normalized intensities that you can process through your multivariate statistical analysis package in the variety of packages that you can use. Again, let's go back a little bit and define what really type of approach you can use. For metabolomics, there are three major approaches, and each of them has advantages, disadvantages. And the three major approaches are targeted analysis, metabolite profiling, and metabolic fingerprinting. And as we talk, targeted analysis is a very important approach if you know more about your biological process and if you already have a hypothesis you want to confirm. Because it's really a quantitative approach that allows you to precisely measure in a high throughput a set of cellular metabolites, which can range from few to few hundred. The disadvantage of that approach is really a limited number of compounds can be measured, and you will never see the compounds you don't have in your target list. So it really will not let you identify novel compounds, compounds you didn't target, or build new hypotheses based on processes that you don't have envisioned for. And another big limitation is the technical one, because to really do a targeted analysis, you need to have a target compound in a pure form, or ideally even a stable isotope labeled or some other isotope label so you can use as internal standards. The other approach that doesn't have this limitation, that really allows you to look at global profile, to look at all the peaks and all the metabolites you can measure with your appropriate techniques, which is it's a global approach. The disadvantage of that is most cases is semi-quantitative approach, because you cannot have all the metabolites available in a cell as a pure standard that you can spike in your samples and quantitate it. So really normally you would normalize your data to a set of internal standards, or maybe even one internal standard like people use in GC, and really present your data as semi-quantitative data. And the other big limitation for this approach is that majority, a large number of your peaks in your chromatograms cannot be identified right away, and many of peaks remains unknown. And also the informatics of the global profiling is quite difficult, because it's you look at large number of metabolites, you need to align, deconvolute a large number of peaks. Many of them are co-eluting. Therefore, it's a medium throughput. But if you still want to look at a global profile, but if you omit the step of identifying all the compounds and just want to look at your profile as a fingerprint, you can use an approach called metabolic fingerprinting and footprinting, which allows you to treat all your profile as a specific, as a pattern. The advantage of this approach is, again, it's global, like metabolite profiling, and it's directly applicable to pattern recognition statistical techniques, but it doesn't involve step of very difficult informatics of compound identification. And again, it's very high throughput. What really makes metabolomics as a challenging technique is that a challenging approach, let's say, because you really want to look at the same time, measure simultaneously a large number of metabolites which can diverge significantly in their properties, in their hydrophobicity, volatility, molecular weight, and so on. And the truth is, it's not a single analytical technique that allows you today to measure all the metabolites at the same time, because of there's so much diverse physical and chemical properties. And therefore, what most people do in metabolomics, and is really adopted approach, that use a variety of techniques, a combination of techniques, to look at subsets of metabolites that you can measure. For example, volatile compounds are compounds that you can make volatile, use a GC-based approaches like GCMS. And for non-volatile compounds, you can use either normal phase or reverse phase HPLC approaches. And depending on what kind of metabolites you can use, there are varieties of techniques that can be used for metabolomics study, which really creates your metabolomics toolbox. And the most common techniques are the FTIR, NMR, mass spectrometry, and different variations of these techniques. And each of those techniques, again, as we discussed before, has certain advantage and certain drawbacks. So let's look at a few techniques that most common use in a metabolomic study. And again, one of them is NMR, nucleic magnetic resonance, which is really very good techniques for metabolomic study. One uses a lot of early studies. It has rapid analysis time, it has high resolution. Again, it's no derivatization needed. The disadvantages, the main disadvantage to NMR is to be low sensitivity, also is improved lately. And the other techniques is FTIR, which is also has a rapid analysis, and similar like NMR, there's no derivatization needed, and you have a complete fingerprint of a sample chemical composition in a single analysis. But also there are some drawbacks and disadvantages. The techniques, for example, it's extremely convoluted spectra, and you have more than one peak per compound, and metabolite identification is very difficult to do with FTIR, and it also requires sample drawing. So what most people use lately in metabolomic study, and majority of metabolomic studies these days, are made using mass spectral based technologies. And depending on the front end of the separation device that you can connect to mass spectrometer, it can be the GCMS, LCMS, and CMS, or gas chromatography, liquid chromatography, or capillary electrophoresis. And all those techniques have very high sensitivity, but depending on your separation techniques, they have also certain pluses and certain minuses, which you can see on the table. And therefore, mass spectrometry techniques, due to the high sensitivity and the first group of metabolites can be measured, is really a method of choice for your metabolomics studies. And depending on really what is your scientific question, what is really your hypothesis, what questions you want to answer, you should select the most appropriate analytical techniques to do that. And really what you need to consider. So what is your sample throughput, how many samples you need to analyze? And what is your instrument scan speed, how fast you can acquire data? And depending really what you want to do a global profiling, or you have a hypothesis you want to confirm, you have a set of target, and so on. So it's up to the researchers to define what is the purpose of metabolomic study before they can choose the techniques you want to use. And depending on these goals, you can pick up the most appropriate technique, as it can be mass spectrometry, or NMR, or GCMS, or LCMS, and so on. And again, as I want to mention here, just keep in mind always that none of the analytical techniques allows you to measure all the metabolites at the same time. And therefore, many techniques are complementary. In most cases, you should use a combination of techniques to get as many metabolites as you want to measure. As is one of the examples, show you the number of metabolites that can be analyzed from urine samples using a variety of techniques. And you see a lot of metabolites can be measured with one or two techniques, but really none of the techniques allow you to measure all the metabolites. This is very important to consider when you decide which techniques you want to use and what is your metabolomic study is about. Okay, so as we talked, there is many steps in the experiment metabolomic study. And each of them has advantages or disadvantages. And each of them can be more or less important to your final output. And so there are studies show, like one of the ASMS meetings and metabolomics workshop was a survey done. What is really the most critical step and the most difficult step and most challenging step in your metabolomic study? And really, where is the most of the attention need to be put to resolve those bottlenecks in the future? And as you see, really two of the biggest challenges for metabolomic study is compound identification and assigning biological significance to your data. And again, and the third one is really data processing and data analysis. So really instrumentation and sample preparation and other steps is not really so critical these days because the techniques have been developed for many, many years now and are quite advanced. But bioinformatics associated with metabolomics, you mean data processing, raw data processing, statistical analysis, data mining, as well as compound identification as a critical step that need to be addressed during your metabolomics study. And so let's go back and see how those challenges can be addressed. And as example, using mass spectrometry-based metabolomics, how can you improve your workflow to really reduce your challenges and overcome those bottlenecks associated with data processing? And as I mentioned, mass spectrometry is a technique of choice these days for your metabolomics analysis. And the reason is, as you see, the mass spectrometry is the most common used technology for metabolite screening due to the early adoption of the GCMS, or gas chromatography mass spectrometry-based techniques as metabolomics approach. And it was used early days for the analysis of plant extracts, metabolomics analysis of plants, and due to the development of very advanced libraries that allows you to identify a large number of compounds quickly, and due to the reproducibility of electron ionization technique. And EI libraries has now been developed by many labs, and they've been shared by many labs, and which just makes it much easier to do a large study and also to compare data generated in different labs. And later, with the advantages of the ultra-high pressure HPLC, which generate a very high level of separation, and combination with mass spec is another technique that most widely used for the metabolomic study. And the advantage of both UHPLC-MS and GCMS is really high sensitivity, high dynamic range, and good separation up front the mass spectrometer. But it's also some disadvantages of mass spec-based methodologies. For example, for GCMS and sometimes for LCMS, a large number of compounds have to be derivatized to be either ionizable in HPLC-MS or to be volatile for GCMS studies. And even in the EI-MS, in many cases, you don't see a pronounced molecular ion, which makes your compound identification challenging. And for the GC, the EI ionization is kind of harsh ionization technique, so in many cases it breaks compounds too much, generates a large number of ions, which is hard to deconvolute and to put a name on a compound. And also a large number of compounds are not very stable in GCMS. But at the same time, GCMS has been widely used. It's the most common used techniques for metabolomics. It has a lot of utility because it allows you to simultaneously measure a large number of compounds from amino acids, organic acids, amines, and so on. And this is an example of the metabolite profiling using the GCMS techniques. And as you see, you can, at the same run, which is about a 50-minute run, you can see organic acids, you can see sugars, you can see amino acids, and mono, ditrisaccharides, and so on. It's a large number of compounds you can see. Also, many of them have to be derivatized in the two-step derivatization that we'll talk later to be able to see it on GCMS. And GCMS is really an generally accepted techniques because it allows you to apply multiple approaches, as we talked before. It allows you to do targeted analysis. It allows you to do metabolite profiling, and it allows you to do metabolic fingerprinting, depending what you do. It's a very versatile technique, and it has very high separation efficiency. So you can, as you see before, you can use a fingerprint that I show an example and use it normally for your metabolic fingerprinting. You can also do untargeted screening for identify unknown compounds. And you can do it at targeted screening to measure, specifically measure known compounds using internal standards, but you can also use it as a targeted, untargeted screening to really quantify metabolites. Not all of them are really have internal standard. You can quantitate them or maybe unknown, but you have a nice transition to be able to measure that. So, and it is also important, it has a high dynamic range, it can be very quantitative if you're using the triple stage quadrupole mass spectrometers for quantitation using MS-MS approach. And again, most of the metabolomic studies done today have been done using full scan EI MS. But lately, due to development of advanced triple quadrupole mass spec, more and more people trying to think about using it as well for the targeted analysis or targeted, non-targeted metabolomics approach using MS-MS approach. And the advantage of the MS-MS approach is it allows you to really increase your sensitivity and improve noise level to the increased specificity of your approach. And really, if you look at how the triple quadrupole mass spectrometer works, it has 3 quadrupoles. One of them allows you to really select your precursor ion, for example, in this ion 319. And then you collide this ion with the inert gas, for example, argon, in your collision cell, and monitor the transition of your ion to a specific daughter ion, which can be measured and quantitated. In this way, as you see from the chromatogram on the right and the left, you can see dramatic increase in the quality of your spectra, almost eliminating noise completely and increasing your sensitivity level. Okay. So, and again, so let's go through the steps. So you take your first ion, you select your ion of choice. Then you react it with the argon gas, and then you measure it in a Q3. And this way, it allows you to really measure multiple compounds in very rapidly in the same time due to the high efficient scan time and your triple quadrupole mass spectrometer, because you can measure multiple ions in multiple conditions and transition in the same experiment. Okay. So let's give you an example. What is really you can do with a MS-MS approach? Like, for example, in a pesticide, cyanobenzyl S-methyl ester in vegetable matrix, you can really dramatically increase your sensitivity and your specificity to be able to measure this compound in a very complex matrix with a high accuracy due to the ability to measure the transition of the molecular ion into daughter ion in triple quadrupole mass spectrometer. And as you see, you can do a very noisy peak to move a very clean, measurable peak and integratable peak at a very, very low level. And there is another example what you can achieve during using the MS-MS approach. Sometimes it's very difficult to separate compounds which are coelute and the same thing are isobaric, have very similar molecular weight, especially in the mass spectrometers that have unit resolution. MS-MS approach allows you to quantitate and separate the ion because due to a unique transition, unique daughter ion that you can select for your molecular ion. An example here is a methoxymated and TMS-derivatized glucose, which generates the same ion of 319.2, and very similar ion generated by indole 3-acetic acid to TMS, which is also TMS-derivatized, which is important plant hormone, and have very similar molecular weight, but have a different transition. You can monitor transition of 319 to 129 or 319 to 202, which gives you 2 separate peaks. And also those compound coelute, you can nicely quantitate them and measure them precisely. So the biggest challenge, if you want to use this approach, is for metabolomics, is it really the ability to generate, to quantitate all this data and to generate a large number of those transitions for hundreds and hundreds of compounds to really determine the specific transition for each of the metabolites and generate those libraries that can be used to generate your method and measure hundreds of compounds in a single experiment. Because it's very time-consuming to generate transition, to select specific transition and best transition for your compounds of choice. And now I will give this microphone to my collaborator, Dwayne Cardona, who will tell you about the approach that we currently use to generate the specific transition in a high-efficient way and generate those compound-specific libraries.
Thank you very much, Dr. Shulev. So when we began our collaboration with UNT, one of our primary goals was to evaluate our products for metabolomics experiments. And we began by asking Dr. Shulev and his team the question, how do you get to your metabolomics results? To understand how each of these pieces fit into the metabolomics workflow, we evaluated each step in the process that he described. The result is displayed here. In our current GCMS metabolomics workflow, we have optimized each step in the common metabolomic experiments. This approach will increase analysis throughput in process bottlenecks that we have identified from our evaluation. In this workflow, we've added some automation to speed things along and removed some of the barriers at arriving at targeted metabolite quantitation earlier. Ultimately, we want to provide a solution that gives our users more time to interact with their data and extract the valuable information from their samples while spending less time in the laboratory. So let's start by looking a little bit closer at the workflow details, starting with sample prep. Here we are using a two-step derivatization procedure to prepare our samples for the GCMS. Using the Tri-Plus RSH autosampler, we have added automation to add accuracy and reduce the time between sample preparation and analysis. Our autosampler is configured with a temperature-controlled drawer for sample storage, an incubator for derivatization, and a vortexer to provide mixing during the various additions of reagents to the sample. After the fully automated derivatization process, the samples will be injected on the instrument. In addition to this, subsequent sample derivatizations can be completed during the analysis of the prior sample. This approach standardizes the amount of time between sample preparation and analysis and limits the introduction of variables from sample prep. Now let's move on to the analysis portion of our workflow. As we identified in the previous slide, one of the key bottlenecks in metabolomic experiments is metabolite identification. Screening experiments must be carried out to identify components of our sample through spectral matching with full-scan EI libraries. As Dr. Shulev mentioned before, this is only half of the puzzle. After identifying the components found in our sample, we can begin our quantitative analysis to measure the concentration of our metabolites. Targeted metabolite quantitation provides some of the most valuable information when assigning biological significance to sample sets. In our workflow, we are using the dual functionality of our TSQ8000 EVO to combine full-scan metabolite screening and targeted quantitation with SRM in a single injection. So in order to optimize the use of our triple quadrupole, we needed to understand and identify some of the common barriers or problems experienced in comprehensive screening and quantitation methods. One key challenge for using a triple quadrupole is the ever-expanding target list of compounds that you're looking at. So new compounds will eventually need to be added to your methodology. Changes in sample condition, changes in organisms' metabolism, and the discovery of new compounds in our profiling evaluations can contribute to the need. For triple quad analysis, this is particularly challenging because the development of SRM transitions for new compounds can be very difficult for new users, and we have an actual slide showing why that is so difficult, or can be so difficult. So shown here in a good bit of more detail are the labor-intensive steps necessary for developing our SRM transitions for our new compounds, potentially. As you can see, the many steps involved in traditional SRM transition development adds unnecessary complexity to the laboratory experiments. And it would also require a large amount of investment in terms of time from your either postdoc or your grad student's time in the lab. So we really want to get away from that and try to spend as much time with our data versus in our lab doing method development. So in response to this need, we developed the software AutoSRM. Using AutoSRM, we are able to quickly develop our SRM transitions for our target metabolites. We can achieve this with an automated walk through the method development process. In this process, we will optimize our SRM transitions that we will use for our metabolite quantitation. Use of the software involves 3 steps in the process shown here. We start with the precursor ion selection from our full scan data. Then we move to product ion selection in step 2. And then finally, we arrive at collision energy optimization for our product ions in step 3. So that's just a brief overview of the AutoSRM process. If we look at it in a little bit more detail on what it's automating, and what makes it so beneficial is that it does automate the entire process of the method development. So after you enter a compound's retention time and vial number in step 1, the software sets up injections to collect full scan data. The result of this is a list of ions found in the full scan spectra and the relative intensities. Often we select the most intense ions from the list and choose them as precursor ions. Now, following selection of your precursor ions, the software will set up another series of injections for product ion evaluation. The result of this is another list of product ions and their relative intensities. After selecting your desired product ions, the software will again set up the injections automatically to optimize the collision energy for each product ion. In this final step, we measure the collision energies, which produce the greatest signal for each product ion, and we arrive at our transition that we need to monitor our compounds. So in 3 seamless steps, we've developed an SRM transition and dramatically reduced the complexity of the development, as well as reducing the time of method development that we're spending in the lab. So now that we have our SRM data, we need to move this info into our existing method. AutoSRM will export all of our new parameters to the data analysis software TraceFinder, where we can perform our data analysis. In TraceFinder, we've created a compound database of metabolites and their SRM transitions that were produced by AutoSRM. This information can be easily transferred into 1, the instrument method that will eventually be used to analyze the samples, and 2, the processing method that will be used to analyze the data. The integration of AutoSRM and TraceFinder eliminates the need for copying this detailed information into multiple locations and reduces the amount of errors just from copying that information alone from place to place. So shown here is a partial list of our Metabolomics CDB that we've been developing. So far, we've developed SRM transitions, including multiple derivatized products for primary amino acids, organic acids, and sugars under a standardized methodology. We're continuing to expand this list using AutoSRM and our workflow to identify new library candidates. Integration of our sample prep automation, universal profiling methodology, and Metabolomics CDB into your experiments will allow simultaneous full scan and SRM data collection from the first sample analyzed. And what this is going to do is just provide you with more time to interact with your data and extract the valuable information that will support your hypothesis eventually. So we have some output from using the Metabolomics CDB on some actual samples, and I'll get Vladimir to come back and lead you through that data analysis.
And as Dwayne mentioned, this is really very useful tool that we found and very efficient way to improve your and speed up your method development and spend more time really on focusing on biological questions. And so we generated this compound database and really tried to use it to profile a set of Arabidopsis sample to see if we can use this approach along with our well-established approach of using full scan and get more quantitative data. And it's really a very good approach. And you see the biggest advantage is that it allows you still use your full scan data and really collect all your full scan information in your existing workflows, and at the same time develop MRM method and use quantitative method to improve your quantitation of the compound of low abundance. And as you see here on the slides, we can see the portion of chromatogram which can be look at the full scan mode, which is in the upper panel, as well as an SRM for some compounds. At the same time, you can access your spectral data from your full scan data acquisition and measure spectrum with your in-house or other libraries you can obtain to identify your compounds. And here's an example. One of the examples of the organic acids is in Trace Finder. It allows you really in a very quick way to review your data. And for example, a succinic acid, you can look at all your compound database, the target list that you have in your method. You can see all the multiple transitions, and the advantage of this, you can have a multiple transition for compounds for both purpose of quantitation. You have a quantitating ion, but you also have your quantifying ions, and you can select the ion depending on your matrix because of the matrix interference. Some ions may not be suitable for a particular matrix. So you have a multiple transition for compounds, makes your workflow much more efficient. At the same time, as you can see, a low panel, you can access the full scan data, and you can look at your spectrum of your compound of interest. And the same, you can, with one click of a mouse, you can go to the NIST software and do your library page. It will give you a score depending on your really library match. And as you see, a very high probability of succinic acid data. And we did it for multiple classes of compounds, including amino acid. Is another example with L-valine. It does the same thing. You can see your SRM transitions with all the data associated with that in the integration. You can also use full scan spectra. You can get your spectra and do the library matching. I can give you a few more examples. There's another example with a glucose 6-phosphate. It's the same thing. You can have a very good quantitation using SRMs. You can use a nice peak and a good spectrum, which can be matched to the NIST libraries. Full scan library can do this. Fructose is another example of important carbohydrates in plants, and you can again see the almost ideal library match for the fructose. And the big advantage also is that you can, at the same time, acquiring your triple quad data and quantitative data for a targeted approach, you can also design your metabolite profiling experiment and do untargeted analysis and really use all the tools that use untargeted analysis before. As an example, using the Sieve software that allows you really compare samples and identify features that can differentiate between your sample using multivariate statistical analysis in a typical non-targeted metabolomic experiment. So there's a direct link from a trace finder from your full scan data in a Sieve software that brings you in a Sieve that allows you to do multiple steps. First of all, it allows you to take your data set, a large number of chromatograms, and use unique chromatogram alignment algorithm within Sieve to align all your data, all your raw files. And after you align all your data, you can really do your statistical analysis. You can do your framing and identify the frames or features that are most significantly different between samples due to multivariate statistical analysis and have a multiple visualization tool. And again, so Sieve is an integrated workflow that allows you process your data set for the non-targeted metabolite profiling experiment all the way from raw data processing into multivariate statistical analysis and data visualization. So it has 3 modules which has a specific purpose. One is really aligning models, which allows you to take a set of chromatograms from your large data set and align them using the scalable adaptive tiled algorithm that allows you to use multi-processing features of your computer to do its very quick alignment of a large data set. And next, as it really allows you to identify features that can be used in multivariate statistical analysis. It's really called framing. You divide all your data set based on retention time or M to Z windows to identify areas of that are significant, and it's quite different from a typical binning experiment because you really only take a frame that have a signal there, eliminating all the frames that has no signal in, and use only this data-rich frames into your multivariate statistical analysis. As the next step, you use multivariate statistical analysis to really compare your data sets to identify features that are most discriminative between different groups. And then the next step is really allows you to take accurate mass feature to identify your compounds through the search through a variety of databases. So it's really a big advantage of this flexible workflow that we developed that allows you to use both the targeted and untargeted approaches, the same experiment, collect two layers of data sets. One is really for targeted quantitative analysis at a very low level. A second is to your typical metabolomic experiment that you really want to identify novel metabolites that may not be in your target list. And this approach can be used in a variety of applications, including industrial application, biofuel development, in your improvement of crops, to identify different compounds associated with stress, resistance, and nutritional qualities. It can be used in pharmaceutical development, industrial material development, so on. So this really makes this approach very interesting and very useful. And advantage of having automated software that really takes away a lot of time and labor that you normally put into method development and let you focus on the data analysis, data interpretation. It's a big advantage of this approach. Okay, thank you very much. I think we can really finish here. Thank you for your attention. And if you have any questions, we'll be happy to answer it.
All right. Thank you, Vladimir and Dwayne. Let's go ahead and proceed to the question and answer segment. So if you haven't yet submitted a question, now is the time to do so by clicking on the Ask a Question box at the bottom of your screen. So let's see what questions have come in so far. First one is, targeted metabolomics usually focuses on a subset of predefined compounds of interest. This is hypothesis-driven approach rather than a hypothesis-generated approach. This way, the chemist can miss quite a few metabolites of significant importance for a metabolic pathway, though. Can you comment on the utility of the GCMS-MS triple quadrupole for this?
So absolutely, I think I can answer this one. So we can absolutely focus on those predefined compounds using the functionality of the triple quads with SRM monitoring on those targets. But the further utility of the triple quad allows us to monitor full scan as well as SRM at the same time. And so the benefit there is maybe there's compounds that we haven't identified yet, those unknown unknowns that might be popping out of the noise that we can later target and use that utility of the triple quad with SRM analysis down the road, but we wouldn't know that they were popping up unless we had monitored in the full scan as well. And so that's a little bit about the utility from the triple quad, as well as being able to do the targeted analysis on those compounds removes a lot of the noise or potential false positives from other compounds that might be coming through at that same retention time. And so we're really focusing on the selectivity of the triple quad to really exploit the identification of those compounds and be able to target them selectively and get reliable data from our system.
All right, our next question. Which system is more suitable for targeted metabolomics experiments? Would that be GCMS or LCMS?
Okay, so with GCMS and LCMS, we can do targeted analysis with both systems. However, with GCMS, we're using a helium carrier gas as our mobile phase, and that limits the influence of our mobile phase on our sample analysis, whereas with LCMS, sometimes you'll see the effects of that mobile phase being a solvent on your sample analysis. So, for example, in ion suppression or matrix contribution, you might see that with your LCMS. Surely we need both techniques to cover entire compound classes that we want. Some LCMS is better for some things, and GCMS is better for some things. And to get the entire compound coverage, we need both of those. But for targeted analysis, we do see the enhancement of using SRM and the resolution, combined with the resolution of GCMS, that really shines through for targeted quantitation and looking at specific compounds.
All right, on to our next question. It says, for non-volatile compounds, how critical is the derivatization step prior to the GCMS analysis?
Okay. So with derivatization, we see a lot of questions from LC users on why we're derivatizing our samples and why is it necessary. So with derivatization, amongst other things, we're increasing the volatility of our samples to get better migration out of the inlet, as well as decreasing the thermal lability so that we're not breaking down those compounds within the inlet. So it's critical that we have that so that we can actually see the compounds that we're looking at, and number two, so that they won't break down in the inlet, but also it aids in the separation of our components from one another in the different degrees of derivatization that we can get. So we're actually able to separate compounds between each other better. Added in our workflow, we have automized that process, and so that was another bottleneck that we were seeing from LC users migrating to GC, was the need for that detailed sample prep. So in our workflow, we fully automated that with our auto sampler, and we really limited the amount of time that you're spending prepping your samples to kind of mitigate that issue.
All right, one of our attendees asked the question, I normally compare the relative concentrations of metabolites in my samples rather than the absolute concentrations, and this is enough for me to see differences between biological samples. So why should I detect the absolute amount of my targets? So
the relative amounts, those can vary depending on matrix and sample conditions. So if we have a matrix that's influencing a compound one way in a sample, it could influence it a different way in a sample and skew the relative amounts of the components within the sample. And so if we look at the absolute results by targeting with SRM transitions, we're able to remove any of that interference that we might see from matrix and or from instrument condition, and looking specifically at those compounds with limited noise. So we can bring the selectivity for those compounds down a large degree so that we can actually see the absolute differences between the compounds versus the relative, which could be skewed by those influences.
All right, our next question is, why should I change from my single quad GCMS to a triple quad GCMS-MS?
All right. With MSMS, we're actually adding selectivity to our analyte detection and our analyte tracking abilities. So we're able to be more specific and in terms, or more selective, and in those terms, add more selectivity to our analysis. So we are actually able to see compounds at lower levels without the influence of other compounds around, or that same retention time with the compound that we're looking at. So with a single quad, we're not able to add that selectivity outside of single ion monitoring, which can be inspecific in that you're not analyzing a specific reaction product. You're only monitoring a specific ion for that spectra of that compound, and that can be skewed from compound overlap within the sample itself. So with the MSMS functionality, we're actually able to target specific reactions for that ion that we are looking for within the spectra of that compound, and the selectivity gains from full scan to MSMS or SIM to MSMS are drastic.
Okay, next question. It says, what is the specific benefit of accurate mass in the metabolomic mass spec experiments?
So I think I'll actually let Vladimir take this question. It's actually, there's two really big benefits of using the accurate mass in metabolomics experiments. One is really allows you to increase your resolution, to really being able to measure the mass precisely allows you to differentiate between compounds that may have very similar mass, accurate mass. And this is a big advantage. As you measure thousands of compounds and hundreds to thousands of compounds and sometimes tens of thousands different ions. To being able to navigate through this really very dense data space makes it much easier if you have an accurate mass. And the second is much more beneficial if you have unknown compounds that you want to try to identify from your sample. This allows you to, based on accurate mass measurements, to determine the elemental composition of your compounds, and along with isotoping patterns, it allows you more precisely to put a target or put a probable compound of names of your compounds and to identify it later. So it really can identify compounds much more precisely using accurate mass. And it's actually more beneficial if you have using atmospheric pressure ionization techniques when you have your molecular ion more predominant or abundant. Then you can really have your molecular ion put elemental composition, have your fragmentation, and with the fragmentation you can really deduce and interpret mass spectra much more precisely using accurate mass.
Okay, I think we have time for one more question. This may be more directed towards Dwayne. The question is, Is the SRM transition library published?
Yeah. So the answer is not right now. We're, like I said in the presentation, we're actively including more compounds and trying to evolve it into a bigger library so we can that we can offer to our customers for use, as well as get out there in the published media so that people can see the work that we've been collaborating with UNT and Dr. Shulayev's lab on. So not currently, but soon we should have that information out there for the metabolomics efforts.
All right. Thank you, Dwayne and Vladimir. It is now time to conclude the question and answer session. I want to let you know that today's webinar has been recorded and will be available for viewing in the next few days. We'll send each of you an email with details of how to access the recorded webinar along with a PDF of the slides and instructions how to customize and print a certificate of attendance. You can also access the certificate of attendance right now from the Resources tab at the bottom of your screen. So this concludes today's webinar, and we look forward to your attendance at future events from Current Protocols. On behalf of our speakers, Vladimir Shulayev and Dwayne Cardona, and from me, Gwen Taylor, and our sponsor, Thermo Fisher Scientific, we hope you've enjoyed today's webinar and learned some valuable information. Thanks so much for attending.